Hello? Oh, that works. Okay, we're actually going to do karaoke. I'm just kidding. Welcome, everybody, to today's seminar. I'm really excited to introduce you all to Adrian Steyer. I've known Adrian for a long time. He was my TA when I was an undergrad taking a field class, like many of you are taking right now. He taught me everything I need to know about fish, and I don't know that much about fish, so sorry. But I am able to tell the difference between a damselfish and a wrasse, and I feel like that's all you. So we met then, and that was a number of years ago, and we ended up seeing each other multiple times over the course of those years when I was in undergrad, and then I joined the same lab that he was in for his PhD right as he was leaving. So it's been super exciting for me to hear about all the things that you've been doing over the past several years since we overlapped for 2 months as PhD students. Since he graduated with his PhD at the University of Florida, he did postdocs at UBC, University of British Columbia, with NOAA in Seattle. I remember we just talked about this. And now he's been a professor at UC Santa Barbara for almost 10 years. His research focuses on resilience in marine ecosystem. He uses a combination of fieldwork, data syntheses, and ecological monitoring, and does, I think, very fantastic marine ecology fieldwork, marine ecology research. His work takes him from kelp forests to the coral reefs of Moorea. And a fun fact that I think he's going to show us a video of is he and his team trained a pufferfish this summer to eat out of their hands. So yeah, get set for some adorableness. So let's welcome Adrian and...
I'll grab that thing. Thanks, Anya. You guys hear me okay? Okay. Well, I'm gonna I told Anya I'd show you a video for fun, so you're gonna get some just like random natural history real quick. But there's these fish in Moorea that I've totally fallen in love with. They're Erythron meleagris, which is a guinea fowl pufferfish, and they're super shy. So we caught a bunch this summer, and then we've been doing a bunch of experiments on coral predation, which I'm not gonna talk about today. But the way these things eat is amazing. So I thought I'd show you a video that my undergraduate, Hayden Vega, put together. This is a little finger of a Acropora coral, and you're gonna see this pufferfish come in and do some damage. So imagine, that eating rocks for dinner. That's the real problem with coral reefs. If we just solve this issue, then we'll be totally fine. Okay, so I'm gonna back up for 1 second and say that I wanna try to answer, or at least posit an answer to the question, is adding habitat enough? And I think this is, in some senses, a simple question that a lot of people think that they know the answer to, but I think it's much more complicated when we start thinking it through. So do ecosystems recover when you add structure, or do the occupant neighborhoods that form around those ecosystems also drive the success of those habitats? I'm gonna talk about coral reefs today, but I think this is true for oyster reefs or seagrass beds or mangrove ecosystems, where the habitat is actually forming the foundation of this ecosystem, and these feedbacks between habitat and occupants can drive the recovery of heterogeneous landscapes. So I'm gonna show you some experiments and some modeling exercises from research we've done in French Polynesia. And I wanna start with a quote that I really like. That quote is, "Pufferfish are cool." No, sorry. Is the acid test of our understanding is not whether we can take ecosystems to bits, but whether we can put them together in practice and make them work. But one of the reasons I like this quote is because I was telling some of the graduate students at lunch that I started my PhD in a program that was about very much like a biology department, basic science, and I didn't really learn about applied science very much until after I did a postdoc with the Fisheries Service. And one of the things that experience taught me is how little we know compared to how much I thought we knew. Meaning, as ecologists, we're often taught to pick a model system to test a given ecological theory that's going to be an effective study system. But in restoration or fisheries management or conservation, we're often presented with a problem where we don't get to pick that ecosystem. And so what I like about this quote is that it's about the interface between the ecology of the system. If we can understand how it works and put it back together, then we potentially can facilitate effective restoration and conservation. And we really need that, whether it's mangroves, coral reefs, kelp forests, oyster reefs, or seagrass. We've seen significant declines over the past century in these ecosystems. And that's prompted restoration to be a global priority. So the UN has declared this the decade of ecosystem restoration. The idea is to prevent, halt, and reverse degradation, to recover biodiversity, and improve human well-being. And this very much puts forefront and center the idea that we have to understand how ecosystems work in order to put them back together. There are some success stories that I want to point to before talking about the challenges associated with restoration. These are real things that have happened in my lifetime, where bald eagles, our national bird, is off the endangered species list. That's because of banning DDT, of specific management action to specific recovery success. The whales that were jumping off the coast today are going off the endangered species list. If you look at a population trend for humpback whales on the West Coast that's estimated by NOAA, it's like classic exponential growth. I mean, it's amazing what happens when you stop whaling. And that's true for halting fisheries on the West Coast as well, where benthic trawling on the West Coast had caused all of these major declines in fisheries, yields that were happening on the West Coast. A ban on trawl fishing for a long time has allowed many rockfish and groundfish species to bounce back. So there is evidence that we can insert ourselves into ecosystems and attempt to facilitate the recovery of specific species or ecosystems. So before going further, I want to talk about this concept of recovery or restoration and be really clear about what I mean by success and failure. And this is the operating definition that I'll use today. So imagine on the y-axis here is the ecosystem structure of function of interest. And on the x-axis here is time. So here you have your system going on its merry way, and then some shock happens to that system, and it experiences some end of its disturbance. And then you have a potential for two different time series here. One of those is the system bounces back to the state that it was in before, and another one is it bounces back some, but is incomplete. So scientists who have been thinking about restoration have tried to conduct meta-analyses or syntheses of restoration projects to see how successful we've been. There's been two of those that I want to show you some core results from for the past 2 decades. One paper by Jones, which synthesized over 240 studies of restoration, found that 30% of studies found no recovery following a big disturbance. 35% found incomplete recovery, and then 30 were found to be recovered. So only about a third of the time when we insert ourselves into facilitating recovery of ecosystems are we successful. Now, those are pretty good odds if you go to the casino, but you can imagine that we might want to do better than that. The second study I found even more interesting by the same research group, where they did another synthesis of restoration projects. And in this paper, what they did was compare the blue and the red here, where they're comparing active restoration to passive restoration. So that's when you either actively insert yourself, maybe you're trying to add new animals, add habitat, remove some process that is causing a problem, or just stopping what's happening, stopping the damage that's happening, stopping that disturbance. That would be passive. And what you're looking at here is recovery completeness on the x-axis. This is a scaled value. But what you can see is in the red and in the blue, you have essentially identical outcomes for how complete a recovery project is when people actively intervene versus when they passively just stop a disturbance. And this, to me, points to active restoration, on average, potentially being no better than passive restoration. So to me, that also points to an issue, which is that we may not know enough about how ecosystems work such that when we intervene, we have a higher probability of success. So there's work to be done there. So today I want to talk about habitat restoration in particular. So habitat restoration in nearshore marine ecosystems is our primary lever for restoration, obviously not the only one. And whether it's oysters or seagrass beds or corals, the logic here is that we add structure that attracts colonists that come from an external larval supply, and that we theoretically will regain that function. This brings me to the Field of Dreams hypothesis. Now, most of you aren't old enough in this room to know this movie, but this is your quick pop culture quiz update. So here's this movie. It stars Kevin Costner, who's still alive today. He's Ray Kinsella. He's an Iowa farmer, and he's down on his luck. He's lost his father. He doesn't know what to do. And then he hears this voice, and it says, "If you build it, they will come." And he keeps hearing this voice over and over again. He does what a good farmer does, and he takes his tractor out, and he plows his entire cornfield down, and then he waits to see what happens. And what happens is a little bit of a ridiculous plot too, where ghosts of baseball's past start coming out of the cornfield and establishing themselves as this new community event location where all the community shows up, and even his dad shows up to talk to as ghosts, and it makes Kevin Costner very happy. So why am I telling you about this? Well, this Field of Dreams movie, gotta go see it, you can watch it probably for free on YouTube, and then is inspired a hypothesis that's been discussed in the conservation literature called the Field of Dreams hypothesis, based on this idea of if you build it, they will come. The meaning of this, if you take it to its extreme, is that if you establish habitat in a given location, that you will have that habitat fill up. And as marine scientists, we think about this a lot. We throw a settlement tile out into the field, it gets covered in stuff, right? You put a cage out, covered in stuff. You gotta go scrub it, right? So it makes some sense that we might anticipate that if we just deploy some new structure, that the ecosystem will emerge. So if we try to put that into numbers or quantify it, we can think about that in terms of abundance or in terms of density. So if this is habitat amount on the x-axis, where you have less habitat and more habitat, if we think about abundance, we can think about our Field of Dreams hypothesis leading to a proportional increase in abundance with an increase in habitat. And what that means is, under this scenario, the density of occupants doesn't change. So look at this as a scenario of generic habitat on the x-axis and propagules coming in, raining down from the sky. So imagine those little dots on the top are larvae. So on the left-hand side, you've got low habitat availability, and then on the right-hand side, you have high habitat availability. In this propagule pool, those larvae are swimming around, looking for a good place to live, and in a simplified scenario, they drop down once they're competent or have run out of their yolk in their egg sac, and they come down to the habitat, and some of them succeed, and some of them fail. So under low habitat availability, in this scenario, we get 10 individuals on the left-hand side, and under high habitat availability, we get 20 on the other side. So in this scenario, you see a doubling in habitat from 2 to 4, and a doubling in the amount of propagules that are coming to the habitat. So this is our Field of Dreams scenario. Here's an alternative way of thinking about this. This is what we're calling the propagule redirection hypothesis. So in this scenario, you have, again, your low and high habitat availability on the x-axis, and again, your propagules are raining down, but instead of the first pattern where you see the same thing going on, where you get 10 individuals to 2 habitats, something different happens. And it's not necessarily just larval limitation, it's also maybe larvae just not going to the right place because they swam the wrong direction or got a wrong cue or just made a mistake. And so here's a scenario where you double the amount of habitat and you only get the same amount of propagules on that habitat. So you're redirecting those individuals or redistributing those individuals among the available habitat. If we draw that out and compare it to our Field of Dreams scenario, where again you have abundance on the top and density on the bottom, in the extreme sense, this redirection scenario, increases in habitat might lead to some increases in the amount of larvae in that scenario, but the density of individuals is declining because you're getting more habitat but fewer individuals there. And so these are two extreme alternative perspectives on how we might think about how adding habitat to ecosystems might interact with the colonization stage of marine organisms to affect the number of individuals that emerge on a given habitat. The second thing I want to talk about this afternoon, is how habitat occupants might interact with habitat restoration or changes in habitat heterogeneity. In particular, we know from lots of studies on a variety of systems that the organisms living inside habitats often interact with that habitat. They might graze on the habitat, there might be organisms that use that habitat that are predators, or they might facilitate that habitat, for example, by recycling nutrients. And so in this talk, what I want to posit is that there is a potential for habitat heterogeneity affecting occupants, which is something we've thought about for a long time. This is just the principle of landscape ecology, where heterogeneous landscapes drive heterogeneity in the occupants, but that the effect of those occupants might also, in turn, feed back to affect heterogeneity, causing changes in that landscape. So I'm going to show you an example of how I think this is potentially emerging in coral reefs. But before doing that, I want to introduce you to our study system. So this is a reef in the South Pacific, and you can see lots and lots of biodiversity here. And I want to zoom in on this green circle on the right-hand side here. This is a coral called Poeciloptera, or cauliflower coral. And this coral doesn't look like it has a lot of occupants inside of it, but I'm going to show you that it's very much alive on the inside. So for those of you who are less familiar, I want to talk briefly about what a coral is. So a coral is an cnidarian, and it has these photosynthesizing dinoflagellates inside of it. And the reason I'm bringing that up is because I'm going to show you how the occupants that live on a coral can feed back and affect the performance of the coral itself. And it's really through this symbiosis and the close proximity of these occupants of a coral interacting with the coral that can affect the performance, growth, and condition of a coral. Okay, so Nat Geo a few years ago did this concept called 1 cubic foot. And they went to Central Park, they went to the desert someplace, and they went to Moorea and a couple of other places, and they took this green square, and they basically scooped everything up, smashed it into pieces, and pulled out every single organism they could find in there to show how biodiverse ecosystems can be. And when they did that with a cauliflower coral, as well as a little bit of other stuff inside there too, they found a wild diversity of organisms inside there. And so this is the ecosystem that I'm going to be talking about today. And inside this ecosystem, there are some things we know and some things we don't know, but there are some occupant-habitat interactions that I want to highlight from research we've done and research other groups have done. One of those is that fish fertilize corals. We know that fish, through excretion of ammonia through their gills or through defecating on corals, can facilitate the growth and condition of corals and even facilitate the capacity of corals to withstand thermal shocks. Here's another example of fish oxygenating corals. If you take a little oxygen probe and you put it into coral at night, especially when no photosynthesis is happening, these little fish, by moving their fins around, are aerating the coral and sending away low oxygen water and allowing oxygenated water to get into the coral. Some of the fish that live inside these corals are also good at defending the coral from algae that grows near the coral that can compete with the coral. There's also these amazing little coral crabs that live inside that are bodyguards and cleaners. They can remove sediment and also deter predators. Okay, I've been asked to turn the audio off for this, and I'm not going to do my David Attenborough voice for you, but Anya is. No, just kidding. So here's what you're seeing is a crown of thorns sea star. Some of you have probably seen this before. These are big, giant sea stars that like to eat coral. They can eat a coral the size of a basketball in a day. And this crown of thorns sea star is crawling over top of this cauliflower coral, this Poeciloptera coral, and underneath you can see this little crab down here. What this sea star is trying to do is just doing the sea star stuff, which is extruding its gut and trying to digest things alive, and this crab is not having it. And so what these crabs do, even though they're itty bitty, is they try to defend the coral from these predators by taking their tiny claws and nipping at the part of the crown of thorns sea star that is more vulnerable. So that's the gut that it's trying to stick out and its little tube feet, which you can see here. And so in a second, you're going to see the crab get aggro with the sea star and fight the good fight. Sometimes these little guys win, other times they get digested whole. But in this scenario, the sea star loses and goes on to feed somewhere else. And so there can be lots and lots of different interesting species interactions. These are all positive effects of organisms that live inside Poecilolepora, but here's a negative one, that's a common one. What you're looking at here is a snail that is an ectoparasite that lives on top of the skeleton, and it leaves this lesion here on the right-hand side. And what it's doing is slurping tissue. It's just sitting there stagnant and slurping tissue off the coral. The coral is replacing that tissue, and it's just a little parasite that's sitting there. But we know that these snails can have effects on the coral's performance. And then, this is the hard part about this system, there's a lot of things we don't understand. If you take one of these corals and you empty it out with some anesthetic, you're gonna find about 50 to 60 different species of fishes and invertebrates, only a fraction of which we know the biology of and barely know the taxonomy of. And so all these little fishes and invertebrates are living inside the coral. It's a total ant farm of species interactions and different species of fishes and invertebrates, and many of which are interacting with the coral. Now back to propagule redirection and this concept by which habitat affects occupants, and then I'll talk more about occupants affecting habitat. So what we did is we looked through the literature and we found some examples of, okay, what do we see in the field in terms of patterns? Well, whether it's the coral number that you increase or the size of a coral, the density of fish per coral tends to decline as you increase the number of corals. And the density of fishes and invertebrates tends to decline as you increase the size of a coral. And so the logic here, or the hypothesis, is that changes in habitat, whether it's the amount of habitat, the number, or the size, can alter the density of these organisms that are living inside, potentially altering the effects of those organisms back on the habitat. Okay, before going further, I want to thank our team, and there's a bunch of people here, so I'm going to talk about our PIs first, so Craig Ozenberg and Scott McKinley, and then Joe Curtis, who was the key lab manager and specialist for this project, 2 graduate students that worked really hard on our project, Alex Primo and Dan Cryan, our Tahitian collaborators, Tahiri and Tarava, and then Elizabeth and Lewis did a bunch of very cool modeling, which I'm going to share some of which under Scott's mentorship, and here's Lexi, who was an undergrad assistant for the project. So I'm real grateful to them and to NSF for our funding. Okay, my talk has 3 parts today. I'm going to try to answer these 3 questions. First, do settlers scale proportionally without a habitat? 2, do habitat-driven settlement patterns persist through time? And 3, if they persist, do feedbacks between occupants and the habitat reshape landscapes? Okay, so we're talking about marine organisms here, and that means we have an adult stage that lays eggs typically. Those eggs then hatch and turn into larvae into the water column, and then they settle back to the reef, often in a new location, almost always, as juveniles or recruits, which completes their life cycle. And so they have this colonization and post-colonization phase. And part of the reason why I'm showing you this is the first part of the talk, I'm going to talk about this colonization phase, this settlement phase. How does changes in habitat availability affect colonization? And so if we can think about this idea of propagule redirection, I can show you this cartoon where imagine you have a single coral by itself and it gets a bunch of settlers, again, kind of falling out of the ether onto it, and you get 5 over here, and then you get the same density of larvae that are dropping down, and rather than getting, you have 3 times the amount of habitat, but you're only getting 1 more larvae than you had over here. And so this is the propagule redirection hypothesis. So to test this hypothesis, we did a habitat manipulation. We took these standardized habitat units, which is essentially a coral epoxied to a cinder block, and we created 15 of these, 12 of these replicate box where you have 2 corals on the left-hand side and 2 corals surrounded by a bunch of habitat on the right-hand side. So 2 versus 12. Here's a little picture of what those reefs look like. And then we replicated this experiment across the North Shore of Moray, and we put these corals in areas that had very little habitat to try to isolate the effects of our habitat treatment compared to surrounding reef. Then what we did is go out every day with a little bottle of anesthetic and a hand net and look for little baby fish inside the coral. And you can barely find them because they're translucent. They have a little bit of patterning, but they're coming out of the plankton where they're often clear, and then they're transitioning into their benthic stage. And so you probably can barely see it here, but there's a tiny little bit of baby fish in here. And so I would take that clove oil and my hand net and grab that fish and collect them. And we found 4 different key species that I'll show you data for today. 2 damselfish species here, and then a wrasse and a goby. Okay, so I'm going to show you two different analyses from this experiment. The first is I'm going to compare the ratio of habitat in the blue circles here to the pink circles over here. So if we found no isolation effect, so no effect of having neighbors, we would expect the ratio of colonists in these habitats to be equal to 1. So on the x-axis, I'm showing you the 4 species, y-axis is this ratio. So what we found was that settlement was highest on isolated reefs. So the blue habitats had more settlers than the pink habitats. So there was an increase in colonization when you didn't have any neighbors. The second contrast I'm going to show you shows you all of the different reefs. So this is a 2 versus 12 contrast. So in this scenario, if we plot the ratio of the turquoise ones to the purple ones, so lots and lots of habitat to 2, our field of dreams expectation, where we had a proportionate increase in colonization with increases in habitat, is at 6, and our redirection effect is down at 1. And what you can see here is that for all of our species, we found very few increases in colonization, despite adding a whole ton of habitat. What we found was a 1.36 increase in the amount of larvae with a 6-fold increase in habitat. Okay, so in this experiment, just focusing on colonization, propagular redirection dominates settlement patterns. So here we found that isolation detracts settlers from focal habitat and that settlement increases disproportionately with high habitat availability. Okay, so on to our second question. I just showed you what's happening at the colonization phase, but one thing that's relatively well studied is the post-colonization phase after a fish gets to the reef. So what happens to a little baby fish when it gets to the reef is it swims in often at night, and when it gets there with all of its little friends, it then tries to find the best place to live. And for fishes that have high densities of individuals when they settle, they have high mortality. And the reason why is that if you look at the distribution of fish across these reefs, what you see is increasing density leads to increasing percent of fish that are living in vulnerable positions. The best competing fish live at the center of the coral, and they kick everybody else out of the house so that they're living on the edge, and those individuals living on the edge get nabbed by predators. Okay, so the reason I'm showing you this is because this is density-dependent mortality, which is a compensatory effect whereby increases in density do not necessarily lead to increases in new individuals. And so what Elizabeth Hammond, who's a postdoc on our project, did was to build a model that explored the potential role of these colonization patterns and the post-colonization patterns and how that drove long-term persistent patterns that went from habitat heterogeneity to the abundance and distribution of individuals at equilibrium. So here's an example of how density dependence might modify a pattern that's initially established at settlement. So here you have the field of dreams scenario where you have 3, 3, and 3 on 3 habitats, and here's a redirection scenario where you have more individuals on your habitat that is more isolated. This, through density dependence, could lead to this pattern post-settlement because the high density of individuals that are living here could experience that compensatory mortality, causing them to die at higher rates at higher densities. Okay, so what Elizabeth did was she built spatially explicit simulations of habitat. So it's a 2D framework, and these little gray dots are little corals. And her landscapes that she built varied from not a lot of variation in neighbors to some random distribution of individuals to a clustered landscape that you can see over here. And so then we simulate the settlement process. And in that process, if you plot the number of fish on a coral and time on the x-axis, what you can see is if you start with a discrete settlement event, then you have continuous survival that happens after it. There's post-settlement survival. And then another occurs, and another, and another, and another, and another, until you get to the point where you have some equilibrium biomass of fish given the separation in timing of settlement arrival and the colonization and post-colonization patterns. So what affects these long-term patterns? Well, there's a few different things. There's density-dependent mortality and density-independent mortality. You can think about that as random variability in mortality that is driven by something that has nothing to do with the density of conspecifics. And density-dependent mortality is going to be what's caused by the density of conspecifics. But there's also the time between arrivals, because we have interacting cohorts of fish that are happening, oftentimes every month during the summer, that are then going to interact with each other. And we've got the magnitude of larval supply. Is this a location that gets lots and lots of larvae, or is this a location that only gets a few larvae? So this model has a few different components. The change in biomass over time is equal to this, where this is the number of colonists, and here is equal to some density independent mortality rate, which is a function of the population size, some density dependent mortality rate, which is a function of how many individuals are there, the input of larvae here, and the time between the arrival of these individuals here. And what this delta function does, which allows us to change the pulse of the input. So what this model does is gives us an opportunity to manipulate the scenarios over which we might see environmental conditions that could modify the patterns initially established by propagule redirection. So here's an example of what a landscape might look like with these redirection effects, where you can see the heat on this plot has settlement in red here when there's lots of settlers and not so many in the purple here. And you can see an isolated habitat has 13 colonists, and ones with lots of neighbors only has 3. Okay, so what we can do is send a bunch of larvae over these landscapes, run the dynamics of these individuals across that landscape, and then look at the model at equilibrium to see what are the circumstances under which we might expect to see the patterns that are established as settlement persist all the way through to what the patterns are that we might see when we go out onto the reef. So I'm going to show you a plot that has a few different panels on here. First is the index of dispersion, and what that means is above this one line here, you have heterogeneous distribution of individuals. You have pattern in your landscape. Below this line, you have homogeneous distribution. You have no pattern in your landscape. And on the x-axis, you have the 3 types of spatial landscapes that I showed you before: even, random, and clustered. Okay, so when we start out with our patterns in our model, what we do is we say, okay, the settlers are randomly distributed across the landscape. Those are little red dots on top of the gray dots. So what we have is no dispersion here at all. Then we have our redirection phenomenon, which leads to increases in heterogeneity across the landscape. That's the distribution of individuals changing because of this neighborhood effect, where larvae are going more in abundance to those isolated patches. Then, under scenarios of low density dependence, what we see is that index of dispersion persists over here. But when we crank up density dependence in this system, that compensatory effect, what we see is that there's very little heterogeneity in these individuals. Because what's happening there is those individuals that settle at high densities on those isolated corals are experiencing really high mortality. So what we can do is to say we've got these 4 parameters in our model, let's try to simplify them down to a simple context-dependent landscape of when we expect propagule redirection to create patterns and when we expect it to just homogenize patterns across the landscape. So if we take the ratio of our density-dependent and density-independent processes and turn them into this parameter, and the ratio of how quickly colonists arrive to how long it takes for ones to die, we can look at this in a 2D landscape or heat map. And what you're looking at here is whether or not the ratio of isolated to clustered habitats is higher than 1. So when it's red, the isolated and cluster habitats have the same number of individuals. And then when you have the blue or yellow, you have isolated habitats that have more heterogeneity across the landscape than habitats that are clustered. And so what you can see here is areas that have strong density dependence, lots of larval supply, tend to have no pattern, and areas that have low density independent mortality or lots of time between arrivals tend to have pattern. And so whether or not you expect the way in which habitats interact with larvae to alter the patterns that you see in nature is very contingent on the life history of the organisms, as well as the landscape over which they're passing and the supply of larvae and how they're interacting with each other after they get there. Okay, so what this means is we get these 2 different patterns of redirection versus field of dreams on this landscape. Up here, we've got our field of dreams phenomenon, and down here, we have patterns that look like redirection. Okay, to summarize this section, what I've shown you so far is that the configuration plus the redirection leads to habitat heterogeneity, or occupant heterogeneity, excuse me, and that 4 parameters determine whether patterns are persistent. Over here where you have pattern, you have low larval supply, low density dependence, and that pattern looks like propagator redirection, and the system isn't really saturated with individuals. But over here, you've got high density dependence, lots of larval supply, and that looks like the system is saturated with no spatial pattern. Okay, on to the third section here. If these patterns are persistent, do feedbacks reshape the landscape? So I wanted to start with a short anecdote. This is different than my movie anecdote, I promise. It's about a title that we had in our discussion section for a paper we wrote. And the editor and the reviewers gave us such a hard time when we wrote our paper about colonization dynamics that they didn't want to hear us talk at all about what that might mean for coral coral occupant interactions. So we, tongue-in-cheek, sent a revision back to the editor called with a discussion section that still kept our text in there, but it said propagator redirection, unbridled conjecture. And they published it. I guess they didn't check it. But I feel really proud of that discussion section. But it also led me to this idea that I want to talk about today for the third component of my talk, which is this idea of dynamic landscapes and a hypothesis that we've built around it. And it's based on the patterns that I've been showing you that have to do with colonization and post-colonization dynamics happening, but that is adding on the idea that the occupants that are living on these habitat, whether it's the fishes or the crabs or shrimps or snails that are living on there, are potentially affecting the growth and survival of the habitat. So let's start out with a scenario where we've got corals that vary in amount, size, and proximity. This is a pattern you would normally see on the reef if you went and swim around. Some little corals, some big corals, some close to each other, some far apart. Imagine then that the coral landscape produces spatial variation in settlement. That's the propagator redirection phenomenon I was just talking to you. Patches that are isolated get more colonists, patches that aren't get fewer. Then imagine that those settlement and post-settlement processes that I just talked about in that model and their survival determine the density of these coral-associated fishes and invertebrates. That's the acronym here, CAFI, that I might slip into. And here you have more individuals on isolated habitats and fewer on these clustered. Okay, if these are organisms that have strong positive effects on the habitat, then this pattern that is established here should lead to variation in how much those corals grow, survive, or have good condition. So here we've got our habitat that's isolated. It's chock full of positively affecting coral occupants. Say it's these fishes that are adding nutrients or oxygenating the coral. And here these little arrows depict increases in the growth of that coral that's higher than the growth rate of some of these corals that have lots of neighbors, where they've experienced that redirection of individuals, they got fewer occupants. And so what's going to happen through time is that if this pattern is persistent, if we close this loop, this asymmetry across the landscape that exists because of habitat-occupant interactions is going to lead to the evolution of the landscape. It's going to change what the landscape actually looks like. And so here, if we have differential growth and survival, that can feed back to actually alter this coral landscape. So the hypothesis here is that rather than having the system be a static landscape that may have experienced some disturbance or fragmentation that then affects just the occupants, is to close the loop and say, okay, the landscape affects the occupants, and now does the occupants come back and affect the landscape? If we take these models that Elizabeth and Lewis have built and play them out by adding interactions between the organisms and the habitat and run them out to equilibrium, and we compare what the patterns of landscapes, again, this is simplified landscapes, might look like under circumstances where you have beneficial coral-associated fishes and invertebrates or deleterious associated fishes and invertebrates, we can see really different patterns. And I'm showing you this for areas that get high coral settlement and areas that have low coral settlement. But what happens when you have deleterious occupants is you have more small, patchy colonies, and the reef exhibits a slower recovery, whereas when you have beneficial occupants, you get larger colonies and higher coral cover. And that can co-vary with the amount of coral colonization that's happening here. But what this is formalizing this hypothesis to say if you have beneficial or deleterious occupants, and they experience redirection as they're engaging with the habitat, then you should see landscapes that look really different in terms of their pattern and their size frequency distribution under these different scenarios. So that's our hypothesis. So here we went out and built habitats that had different amounts of corals on them. And these are really simple, again, similar to the last experiment where we've got these cinder blocks and we strap them down. We collect corals that are all very similar in size. We take this epoxy here, Z-spar, and glue the corals down to that area. And we have this little plate that allows us to take the corals on and off. So these corals, when you collect them, are chock full of stuff. So what we have to do is first collect them and empty them. And you can take the corals and dip it really quickly in clove oil and then use forceps to pick everything out of there to try to empty all the corals. So we're starting out with a blank landscape in terms of occupants. And then we constructed these reefs with 1, 3, and 6 corals. And we did photogrammetry, which is basically taking a camera 2D photo of these corals and taking a few hundred photos of each coral to get a really nice 3-dimensional model of our corals that you can see spinning around on the upper right-hand side there. We then had allowed for communities to assemble for 2 years on these habitats, maintaining our experiment. And then we came back and we sampled all these individuals by using a combination of visual census and then even taking all the corals into the lab and plucking every little last thing out. And the idea is that we can look at these patterns to see, do we see evidence for, over the longer time period, heterogeneity across these habitats that are driven by the landscape, but also to try to link the corals to those communities. So what we see here is that at the abundance of the whole community level, the communities followed the field of dreams hypothesis. Meaning if this here is the expectation based on a proportional increase from 1 to 6 corals, there's no difference between the total community abundance and the number of individuals that you see. So no changes in density that we observed across the entire community. When we look at diversity, we see almost a tripling in diversity that happens when you go from 1 to 6 corals. And even when you control for differences in abundance using rarefaction, you still see that those larger reefs do have higher diversity. So this is good news in my mind. You add more habitat, you get more individuals, you get higher biodiversity. The story becomes a little bit more nuanced when you start to look at what happens to individual species. So I'm showing you this same type of plot, where on the x-axis we've got our coral number, and on the y-axis we've got the number of individuals, and you might recognize some of these species from the previous experiment. And here what we see is some of our species don't follow the field of dreams hypothesis, they instead follow the redirection hypothesis. And this is consistent with the settlement patterns we saw in our initial experiment. Other species, like this predatory snail that I showed you earlier, proportional increases in abundance with increases in the amount of habitat. And then third here, you have species that were barely abundant on these 1 and 3 habitats and then skyrocketed up to be very abundant on some of the larger habitats. So different things happening in different reef habitats. Overall, the total community increased, but certain species were responding differently to that habitat heterogeneity. So if we look at this in multivariate space, we can see that the solitary reefs differed in community composition than the others. You can't see the hulls here, but the green dots here are the individuals that are in the 6s, then the 1s are over here, and the 3s are in the middle. And so if you do a community composition analysis, whether it's PCA or Permanova, you see big changes in the species composition amongst these habitats. And the biggest changes are between the 1 and the 6 habitats. And these are a handful of species, some of which there were more on the 6s versus the 1s. And here are some of the 1s having more than the 6s. And I don't expect all of y'all to be familiar with all these species names, but I can tell you that some of these species that are in here that are on the singletons more abundant than on the 6s are ones that we know of as having particularly strong positive effects on the coral. Okay, so what happened to the corals when we looked at these? We took little samples of these corals, we analyzed them for zooxanthellae density, for carbohydrates, proteins, tissue biomass, and then measured using the photogrammetry how much they grew. And we plopped that into a principal component analysis, and all of those things load really heavily on the first principal component. You can see those loading here on the left-hand plot here. So that's PC1. And if you look at PC1 across our treatments, you can see the individuals where there were 6 corals on average did significantly worse than the individuals that were solitary. Okay, so the communities were different on the singletons versus the larger reefs, and the corals underperformed on the larger reefs compared to the singletons. So what if we smash those 2 data sets together? So here I'm showing the principal component of the community space on the x-axis here, and the principal component of the coral on the y-axis here. And here are the loadings for the communities, sorry. And so again, here the colors correspond to the number of corals. And what you're seeing here is over here, you've got individuals that are in the singletons having high coral condition and a key community composition that differs from the individuals over here that have low coral condition and unique community composition. So here's these differences in species composition that are differing between the singletons and the sextuplets, and here's how that's correlated to the coral. So here I'm showing you that there is a correlation between changes in community composition that happen when you add lots of habitat and the performance of the coral, where the performance of the coral is going down when there's more habitats next to each other. And we can look at this in bivariate space too to make it a little easier. So I pulled out a few correlations here. Here's a coral croucher species that lives at the very base of the coral. Higher abundance of these guys, higher performance. These guys in opposite, these fluffy little crabs here, are negatively correlated to coral performance. So what this allows us to do is to form some hypotheses about which species that we might not have known particularly a lot about might be negatively or positively interacting with the coral. These guys in particular, these Harpiliopsis shrimp, are crazy abundant inside these corals, and we just haven't known anything about the ecology of them, but they are positively correlated with the performance of the coral. So we're learning a little bit about how individual species might be contributing to the performance of these corals and how the landscape might be modifying that. Okay, so for this section of the talk, what have I shown you? I've shown you that community abundance scaled proportionally with coral number. So that's that field of dream response at the community level, but individual species responded uniquely. I showed you that coral number drove key changes in fish and invertebrate density as well as species composition. And I showed you that coral performance declined as coral number increased. And lastly, that fish and invertebrate composition predicted variation in coral performance. And there's two key implications of these findings to me. One, I think we have to continue expanding the concept of meta-organisms. Corals have been foundational species for studying the concept of symbiosis for a long time, but that's mostly been done under the hood, thinking about the communities that are microorganisms and early on only just the dinoflagellates that were living inside the coral. That has evolved to much more incorporate these species here, the microbial communities, viruses, and small fungi that also live associated with the coral and inside the coral tissue. But what we haven't done very well is incorporating in these fish and invertebrate communities that live particularly close with the coral that we know are eating or defending or supplying nutrients to the coral. And I think our definition of how this organism works is really a meta-organism where these species are all interacting with each other together. So there's a lot more work to be done there. Second, I've shown preliminary evidence that landscapes and communities are co-constructed. I showed you that these heterogeneity in landscapes that we experimentally induced caused patterns of species composition that had consequences for the growth of these individuals, where colonies that are living in more large clusters had lower performance than individuals. And if we play that out over the long term, that suggests that we'll see the landscape shift where solitary corals should grow very big relative to individuals that are living in clusters. So I think we've at least attempted to answer these three questions here so far. Do settlers scale proportionally with added habitat? No. We saw strong evidence at the colonization level for propagular redirection, the redistribution of larvae rather than the proportional response of larvae. Do habitat-driven settlement patterns persist through time? Sometimes. Our model shows you that there are certain life history characteristics or ecosystem characteristics by which you would expect to see patterns established at colonization persist over long periods of time, whereas in other circumstances, post-colonization interactions can totally alleviate any patterns established at settlement. And third, if these patterns persist, do feedbacks reshape landscapes? What I think our experiment shows here is that they can. There's evidence in our data set that suggests that habitats where there's more habitat have unique community composition and that influences the performance of the coral. So I think that this has implications for other ecosystems that many of you are working on. For example, here's oyster reefs on the East Coast. So this is work out of Joel Foudry's group that shows significant redirection in oyster reef habitats. So let me show you how that works. Here's Joel borrowing a similar experiment from what we were doing, where we had 2 habitats alone and then 2 habitats surrounded by a bunch of other individuals. And then he followed juvenile stone crabs, which eat oysters, and looked to see through time if there was a difference in the accumulation of those individuals. And here's more evidence for redirection. Here is the high habitat availability area, and the density is way lower over here than in the low habitat availability area. So here's some evidence, at least, that just adding habitat doesn't necessarily give you more crabs. Well, if these are oyster predators, that might mean that those reefs that have lots that are small and isolated, these ones over here that have lots of crabs on them, might lead to particularly high predation, which might modify the capacity of those oyster reefs to persist. And I can imagine this same thing might occur in heterogeneous seascape landscapes, where the organisms that are involved either grazing directly on or removing epiphytes from seagrass habitats might change in their proportional abundance or their composition across these heterogeneous seagrass seascapes, and that might affect the performance or the probability of persistence of these individuals. Same goes for kelp forest ecosystems, where patchiness in kelp can lead to patchiness in the abundance of fishes that are important at recycling nutrients, particularly during relaxation, and that could cause new patterns to emerge in the landscape through time. So I started with this question about, is adding habitat enough? And what I want you to take home from what I've shown you today is that I think we have to keep building in degraded places. I'm not saying anything against habitat restoration. I think it's very useful. But I think we have to prioritize spots that have good larval supply and connections with other habitats. And we have to think a little bit more deeply about how heterogeneity or the spatial configuration of how we distribute habitats can alter those occupants that might strongly interact with the habitat, and let those two-way feedbacks between organisms, their habitat, do some of the heavy lifting. Thank you, and I'll take any questions you'll have.
Thank you, Adrian. Okay, we'll take some questions from the audience, and then if we have some time, take some questions from online. Okay, first question. Way to put your hand up quick. Ask you to talk into the mic so folks online can hear.
Hi. So first of all, I thought this was very interesting. Thank you so much for coming. And I just wanted to say you were looking at primarily the cauliflower corals, correct? And I was wondering if you would expect to see differences in results if you had incorporated different species of coral when you were planting them, and if that heterogeneity would then lead to more diversity or density in the polyps or the just in the ecosystem's diversity.
Yeah, thanks. So yeah, so the question's about whether different species of coral might lead to different patterns in terms of biodiversity or patterns of the occupants. Is that right? Okay, cool. Yeah, so definitely. I mean, there's been some cool experiments that have manipulated the diversity of corals on a habitat and looked to see what fish communities assemble there. And more habitat diversity leads to higher biodiversity and differences in community composite of fishes. So I would expect that. I think in general, I mean, this is a particularly diverse system, these cauliflower corals and everything that's inside there, because it's such a rugose habitat, but the same pattern, I think, is likely to emerge when you have heterogeneity in habitat. And that comes from my perspective about how fish are colonizing reefs, which is like reefs are sending out DMS or some other compound that fish are smelling for at night. And then if they get within this halo or stream of signal that's coming off the coral based on how water is flowing, they're going to swim over to that habitat. And so when we think through the logic of that, we typically end up in a situation where we think, oh, more clustered habitats potentially can capture more individuals, but there's still the potential for lots of individuals for not being able to colonize those reefs. And so that's my guess, is that we'd see similar patterns, but there's a whole lot going on here that still needs to be unpacked. Like fish also smell for conspecifics, and so that can lead to different colonization patterns that can emerge based on priority effects or things like that. So I would expect slightly different patterns in terms of who's living there, because different fish like different corals, but I think I would hypothesize that you'd see similar patterns in terms of isolated patches getting more beneficial fishes and that leading to higher growth.
wants to go next? You guys are pointing at each other. It's really fun.
Spider-Man gif.
Yeah, thanks for the very cool talk. I was wondering if you think the redirection, is this like a short-term initial response to how you put the habitat out, or is like you would expect when you get to kind of the equilibrium state, would you still expect there to not be an increase in density? And if you wouldn't see an increase in density, then what is the limiting factor on the total number of the adults that end up in the community?
Yeah, I think from the way we've been thinking about it, I think you can, let's say it this way. Our model predicts there's circumstances that you can have persistent long-term patterns that go way beyond the colonization patterns that emerge from redirection. That happens when you have low larval supply and weak density dependence. So if you look at like the distribution of density dependence in fishes, some of them are really antagonistic to each other, others are happy to live with other individuals. So I think they're within that realm of potentially having those persistent patterns. But our model also shows that if you have really strong competition afterwards, you can totally wipe out these patterns and not see that. And it seems like from our longer term field experiment that I showed at the end there, that we're seeing a little bit of both depending on the species that are colonizing. So I think it's somewhere in the middle. And of course we could have run our experiment for longer. Just gotta get a little more money from NSF, which is super easy these days. Thanks.
Womp womp.
I'm here to lift it up.
This could be like totally by chance, but I noticed that in your beneficial CAFI, is that your acronym?
Yeah.
You chose the damselfish, and the non-beneficial, you chose the snail, and the damselfish had a redirection type response, and the snail had the Field of Dreams response. I'm wondering if you saw that across other beneficial CAFI versus non-beneficial CAFI, and then like follow-up question, if so, what would that mean for some of your findings?
Yeah, good question. So we see a lot of heterogeneity. There's like all three circumstances. You see like you add habitat, and there's no change in the abundance of those animals. Then we see circumstances where you see like add habitat, proportional increase, add habitat, proportional increase, and then some circumstances where you add habitat and nothing happens, and then all of a sudden you add more habitat and there's this crazy response. And one thing that's challenging to me about this system is that there's so little that we know about the life history of some of our beneficial versus deleterious symbionts that it's hard to plot like a ratio across the landscape of good guys versus bad guys, or whatever, however you want to put it. But we do see a lot of heterogeneity in that, and the hypothesis, at least, that we have is that it's something about the ratio of those organisms changing that's leading to the poor performance of the corals that are clustered, but there's other alternative hypotheses that we have too for why we saw that pattern.
No? Yeah.
No, yeah,
okay,
you're
good.
good.
So I think just to follow up on what Ben was saying earlier, I think if I have this right, so the idea is that the sort of presumption is that essentially recruitment is limiting, or larval supply is limiting to get the results that you've sort of described. Or at least larval supply is more limiting than habitat is. Is that fair?
I think it's fair. The like nuance that I would add to that, and maybe, yeah, I'll add just a tiny bit of nuance, which is that I think you can get redirection phenomenon even if you have lots of larvae in the water column, but they don't find a habitat. So like, just
Sure.
because, that's a little bit of like picking at it. But yes, in general, I think you're gonna get persistent redirection when you have lower larval supply and when there's weak competition.
And so I guess and that sort of fits with what, you and many others have thought about reef fish, right? For example, they are generally supply limited at some level-ish, as opposed to, say, other types of ecosystems. And I guess maybe I'm trying to sort of push to like, okay, if you think that larval supply and or the strength of density dependence would shift you from the sort of redirection to the Field of Dreams kind of hypothesis. Is there anything you can predict about where do you think Field of Dreams will dominate over redirection, like in terms of types of ecosystems, or temperate versus tropical, or invertebrate versus fish, or I don't know, are there
Yeah.
any generalizations you can sort of pop off, or hypotheses maybe I should say, rather than generalizations?
Yeah, I'm gonna pop off. No, yeah, good question. Thanks. I was standing on where the intake pipe is this morning, just like staring at the intertidal, thinking about some papers by Connolly and Roughgarden, which were focused on, for those of you who aren't familiar, like focused on when and where do you see larval supply dominating patterns of what you see in the intertidal, and when and where do you see competition driving patterns. And so in areas that have low larval supply, competition is weak, and where areas that have high larval supply, then competition can be a really dominant process. It's like a really important paper, at least in my opinion, about how we think about context dependency of what's driving assembly of the intertidal and other ecosystems. And I think that that fits the same hypothesis I would have about where I would expect restoration to drive a field of dreams phenomenon. You go somewhere where there's gobs of larvae and those individuals aren't competing very much, then you're gonna get chock full reef of all sorts of stuff. And then you go somewhere else where there's just like a little bit of larvae and there's strong competition, then you're gonna get kind of patchiness across those reefs. And so, you know, this is hand wavy a little bit, but I think picking, like doing little assays before you do restoration and seeing what kind of larval supply you're getting in a given location, or knowing a little bit about your critters and thinking about how much they're competing for space could offer some preliminary insight into site selection for thinking about doing restoration. Does that answer your question? Yeah. Okay. Test.
Shouldn't have teased you. Thank you guys for being here. And Adrian, again, thank you so much. This was great.
Thank you.